on 29th March, Saturday. With this, I would like to request our GK Panda Director Sir of Mid School of Biotechnology to welcome to the August gathering. Sir, please. Sir, please unmute.
Yeah. Thank you. Thank you, Dr. Subratkar. Namaskar to all. On behalf of the organizing team and Mid School of Biotechnology, I sincerely welcome all participants, the delegates, resource persons, and our team management, uh, Chief Executive in absentia, Mr. Siram Panda sir, Mr. Bishwit Panda, the Vice Chairman of our MITS group of institutions, respective faculty members of MITS group, esteemed participants, honored faculty members of different institutes, and invited participants. I take the privilege again to welcome to this allied forum. This FDP is being designed for 5 days, that is from today to 29th, with the theme on next generation molecular diagnostics for pathogens and cancer detection. I believe in these coming 5 days, we will get a forum to discuss, contribute, put our views, and can come up with an ethical research aspects that will be useful for all the faculty members of this biotechnology forum. My sincere gratitude to team management for their invaluable support and encouragement in academic advancements that we have been taking care in our institute. This FDP will engage with experts, explore the latest advancements, and discuss their implications in healthcare and research. I encourage each of you to actively participate, share insights, and make the most of this learning opportunity. Being an IT professional and having enriched experience in artificial intelligence, I personally involved in coming with some of the public in this healthcare domain. Not only biotechnology, with the allied verticals of IT as well as agriculture, micro-agriculture, and all the sectors, I think this topic will give the justice in coming 5 days of time. Once again, I extend my warmest welcome to all participant faculty members from different reputed colleges and universities, and I look forward to stimulating and enriching the session ahead. In fact, there were 94 number of faculty members who have been registered, but as today this +3 examinations has started, a number of faculty members are requesting us to provide or extend some more time so that they can hand over their answer sheets at the examination section and come up. I think we will extend this activity, joining activity throughout the session. And in coming time, I think all 94 participants will go with us with a moment to enjoy this forum. And before leaving, I would like to request all to join physically on coming Saturday to felicitate our resource persons in our MITS School of Biotechnology campus. Thank you once again, and I welcome you all to have a good 5 days of time in this forum. Thank you. Over to Dr. Subratkar.
Thank you very much, Director Sir, for your words. And I am also expecting that the numbers should be going more because the examination is going on, and the maximum faculties which are in the examination duty, they want to join after the completion of the examinations. Hopefully they will join, sir. Thank you, sir. Then now may I request our esteemed Vice Chairman, sir, MITS Group, Mr. Biswajit Panda, sir, to share his keynotes, sir.
Uh, Good morning, everyone. Thank you, Dr. Subrat. First of all, a big congratulations to the organizing team, Department of Biotechnology, Mexico of Biotechnology. Like you have selected a topic which is very important in today's age. The topic is very close to us because we as a family have lost 3 people in cancer within a span of 5-6 years, and very close one. So this is a very important topic. If generally, if I say, like, cancer generally used to be a very difficult word. Like, it's used to listen it very rarely. But when we visited with a mother to a cancer hospital, then we found that cancer is very common. Like, you find thousands of patients with cancer. Like, the most important thing when you go and see the hospitals of a cancer hospital, the most important thing is detection. See, always there is a say, like, if you would have detected early, then probably there are chances of survival is very high. So detection is the most important aspect in terms of cancer and prevention, and easy detection is the most important aspect. So it's a wonderful topic where the team has chosen, like, next generation molecular diagnostic for pathogen and cancer detection. I hope this 5 days FDP will help us in a what do you call, in a brief way to understand, like, how uh, things will evolve in future, and how things will develop in future for cancer detection, or developing, what do you call, molecular diagnostics for cancer. So definitely this 5 days program will be helpful to understand the process in a much more detailed way. And I expect that all the invited speaker, Dr. Sanatan Maji and all, will guide the entire FDP process in such a systematic way that after the 5 days will be a little bit a wonderful brainstorming session among us. And after 5 days, we expect that we will be coming out some kind of outcome, so that for the next level research or for the next level advancement, it will be helpful to us. And yes, this is a good initiative by School of Biology team, that online FDP. And I will suggest Director MSB also to see that you are accommodating all the participants who have registered. If possible, if you can pick the time, or as per the convenient of the entire participant. So, like, let's all people be there. And I expect I definitely will invite all the people who have joined from other campuses, other colleges, to attend on 29th. We will be hosting a small event on 29th, where we will be feliciting the invited speaker, and we'll be distributing the certificates also for FDP participant certificate. And most probably you can join for a lunch too for us. Then, most important in today's age, you understand online, you are connected across the world, but physical presence is little bit connects us more than online, definitely. So I hope that this 5 days will be a little bit excited for us, and it will be, what you call, little like, it should give us some insight about the topic and a brainstorming, so that next level advancement of this process or this topic should carry on. We should carry on with this next level. So if there's all the support from the management to the team, so that anything where we need, we will be there. And we see that this kind of program is happening in the next future, so that we will be collaborating with other institutes, other campuses, other researchers, other people across the country, to see that we are coming up with a good solution as a biotechnology institute, where we might be able to contribute a little bit or more for this cancer detection, or about this topic. So over to you, Dr. Subrat. Thank you. Thank you everyone for participating in the FDP program.
Thank you so much, sir. Thank you so much. and for your insightful thoughts and words. Uh, Thank you, our patrons, for your valuable times and words. Now, I am closing this session. I'll request all the participants to stay with for the days of today's sessions. Dr. Maji, Dr. Sanatan Maji is also is connected with us, and I am requesting Dr. Sanatan Maji to start the sessions. Can we start the session, sir?
Okay, very good morning. Am I audible?
Yes, sir. You're audible, sir. Sir,
Okay, so okay, anyhow, we'll start. We'll
thank you,
start.
sir. Thank you for your consent. Before we
Okay.
start this program, I want to introduce yourself, sir. It is my pleasure to introduce our today's invited speaker, Dr. Sanatan Maji, has accomplished senior faculty in biotechnology department at Utkal University. With a strong academic foundation, he holds a PhD from ICR IVRI institution, where he developed label-free biosensor assay for Brucella antibody detections, and master in veterinary science in molecular virology, and bachelor in veterinary science from OU to Bhubaneswar. Since joining Utkal Institute in 2015, Dr. Maji has mentored 3 PhD, 2 MPhil, and 43 MSc student projects, contributing significantly to biotechnology research. His work includes 17 publications, 18 gene bank submissions, a book chapter, and over 30 conference presentations. He also contributes all an editorial board members for 12 journals and an active member of 9 professional bodies. Recognized with multiple awards of academic and research excellence, Dr. Maji's expertise in molecular diagnostic and biosensing technologies continues to drive impactful research. We are honored to have him with us today and look forward to his valuable insights. Over to Dr. Maji, sir. Sir,
Okay, let me try. Actually, I'm having some technical problem with my this one. Just a minute, okay?
okay, sir.
Because मैंने अपन को screen ये जो इधर का लाइट ऑफ कर दो आंसर
There is some sort of technical difficulty with Dr. Sanatan Maji. He is joining soon. Please stay with us.
Dr. Subrat, can I send my this one PowerPoint to you and Can you
Yes,
play
sir.
from there?
Yes, sir. You can share that one. We
Okay.
will do that one.
Okay. Okay. Totally.
Sir, please hide that one. Sir. Hide button. Hide button. Hmm. No. Hide button. Meet google.com, sir.
Okay. It coming? The screen is coming?
Yes, sir. Yes, sir. It is visible, sir.
Okay. It is visible, na?
Yes, sir.
Okay. So can you start, right? Okay.
Yes, sir.
You can start, no?
Yes, sir. Thank you. You can
Okay,
start.
right, right. Okay. So at the outset, let me thank the chief patron and director School of Biotechnology, MITS, and other faculty members from MITS for inviting me for such a beautiful occasion of today's seminar, that is on the next generation of the molecular diagnostics for pathogens and cancer detections. So itself, I have kept the same topic like that is there in the faculty development program website. So we'll try to understand the meaning of these terms, like what are the next generations and what are the molecular diagnostics mostly adopted for diagnosing the vulnerable pathogens, or as such you can say the dangerous pathogens which are responsible for causing any kind of diseases in our society as a whole, and which are also related to cancer in today's world. So let's see what are pathogens. So pathogens mostly they are regarded as any organisms, may it be a tiny organisms or may it be a little bit bigger organism, but which are responsible for causing any kind of diseases in human beings or an individual, you can say, like bacteria, fungus, protozoa, parasites, and so on, so on. So mostly we'll be dealing with like things like bacteria, which are responsible for causing your cancers and mostly the viruses or prions which are responsible for causing this kind of diseases. Then coming to the second, this one, entity, that is cancer. So a disease which is characterized by uncontrolled cell growth and division, which leads later on towards abnormal cells and that can invade surrounding tissues and potentially spread to the other parts of the body. So more or less we are having a little bit idea on what cancers are like. So it's, you can say that it's a growth which happens in our body, but we do not know in which place that is going to occur. So maybe that is metastatic in nature or that is benign in nature. That depends on the nature of the cancers. But what this definition says is that anyhow there will be uncontrolled fashions of the growth in our body of the cancer cells. But if it get metastatized, so it will travel from one place to another place. So we never know what is the origin of the cancers in our body. But more or less today only we'll be dealing with the cancers which are caused because of the agents like viruses or bacteria or fungus or any protozoas. So broadly, if we go to the causes of the cancers, then we can have these 5 or 6 most broad categories. Like the causes may be chemicals, the causes may be diet, or maybe lack of exercise, maybe infections, radiations, exposure to radiations, hazardous compounds, maybe it is due to the hereditary, or maybe it is due to the hormonal imbalance in our body. So these are some of the causes in pictorial form I have depicted over here. Then what do we mean by molecular diagnostics? As such, we know that diagnostics are nothing but the tools or the techniques which are used to diagnose or to detect the presence or absence of pathogen in the human body or in individual animals. So in that context, molecular diagnostics mostly these are the techniques or the laboratory techniques or the methods which identifies the presence or absence of diseases, thereby detecting the molecules like DNA, RNA, or proteins in the tissue or the body fluids. So based upon that, we'll be discussing on one by one, slide by slide. So what are the importance of today's seminar or the today's faculty development program? Because we know that today probably all faculty from different college or different institutions might be taking participates in this faculty development program. So our duty is to translate the knowledge that we are gathering from or we are gaining from different sources, may it be from different higher sources or may it be from society or may it be from hospitals or anywhere. So that is our primary duty. So to do that, we personally we will have to be so thorough in all these technical aspects, may it be practically or may it be theoretically, but that knowledge has to be translated to the common society at the end. Students will be translated these particular kinds of techniques of detection of the pathogens by doing practicals, by taking those students to the exposure visits to different hospitals of the research institutes like ILS in Bhubaneswar and RMRC, and those knowledge will be gradually translated into the society at the end. So let's see. In picture, I have depicted some of the oncoviruses or the pathogens which are mostly responsible for causing cancer in our body. So pathogen which cause cancer mostly, I have taken this from a paper of Anand et al. 2018. They have shown that worldwide approximately 18% of the cancer deaths are only because of the infectious diseases caused by pathogens, or the cancers which are mostly caused by pathogens. So 18% of the cases have been shown in this particular paper. That means we can say that mostly pathogens or the infectious agent, they are responsible for causing cancers, and those cancerous patients, they are going to die anyhow. That's why they say. So more or less it is 18%. If we see the pathogens or the causal agent of the pathogens, that these are the group of viruses mostly involved in causing cancers in living tissues of the human beings. Human papillomavirus, responsible for causing cervical cancer, Epstein-Barr virus, Kaposi sarcoma herpes virus, hepatitis B and C virus. So mostly we understood that whenever the doctor says us to take vaccines, then we only think about the disease only, but that may lead towards a cancer, and it's very hard to get a treatment for cancer. All of you know, either surgery will be the final destinations, or you can say some other treatments are there, but which may or may not be beneficial for our human beings. So human T cell leukemia virus, one more very important virus which is responsible for causing cancers in human beings. So this data that I have given mostly looking into the human health in concern. So bacteria, if you see the bacteria which mostly involved in causing your cancers are like Helicobacter pylori, or you can say H. pylori, which induces gastric cancer. Then one, all of you are acquainted with E. Escherichia coli or E. coli, that it produces certain toxins. Among the toxins, colibactin is one of the genotoxins which are associated with causing cancers. Now we'll go by one slide so that we can understand the concept or the mechanisms how the cancer develops inside our body. Though there are some microbiota or microorganisms which are considered to be normal microflora of the body. So they are just like, you can say, mutualistic, or they are commensals, or they are symbiotic associated in our body. So how sometimes they behave in a different context or different ways, so that they gradually they cause cancer in our body. So if you look to the parasites, there are so many parasites, but out of all these pathogens, I have taken few pathogens which are mostly responsible for causing all kinds of cancer. If you see, say for example, if you see the cancers of the prostate cancer or cancer of the gallbladder, then they are directly or indirectly involved with any of these pathogens. May it be viruses, may it be bacteria, may it be parasites. We never know what is going inside our body. It's very different or very complex mechanisms going on inside our body. So if you see the parasites, then parasites, helminths are responsible for causing diseases or for causing cancerous. Nematoda, they are also responsible. Nematoda, they are also responsible for causing cancers. But out of them, these two or three, mostly they are prevalent. They are mostly found in most of the cancers. Schistosoma hematobiome and liver fluids, which are responsible for causing, fluids in the liver. So if you look at this particular diagram, then we can understand the biology or the world which are around the cancer. That is, see this the inner cycle who depicts the cancers of the unknown primary or COP. That means we never know that what is the cause of the cancer inside our body, or where the cancerous cells are originating in our body. Or any machines cannot detect these things. It can detect metastatic phenomena. It can detect the final form of the cancer. But what is the cause of the cancer is very, very difficult to detect. So for that, in today's seminar, we are going to discuss a little bit about the different diagnostic tools and different diagnostic techniques, or you can say assays, which can be done in our day-to-day basis in our laboratories also, just to make people aware about the diagnostic process on the cancers. So there are a few things like you can see liquid biopsies and bio nanotechnology, and based upon that, PET and CT imaging. These are the physical methods of diagnosis of cancer tissues inside our body, and this is one of the recent or most advanced form of diagnosing the cancers. And then liquid biopsy is a conventional method of diagnosis of the cancers. Like we have to take the blood samples from the suspected individuals, and those samples will be analyzed in the laboratory to find out the presence of the pathogens or find out the presence of the biomarkers of the cancers. So now let's have an overview of molecular diagnostic. What this molecular diagnostic means actually. So any diagnostics which are based upon biomolecules in our body, just like your DNA, RNA, or any proteins, which are just a building block of our body. So they have to be involved in analyzing a presence or absence of a disease, and they also offers identification of the pathogens and cancer types, enhancing the treatment strategy. So that means based upon the presence or absence of a protein or DNA or RNA molecules, we can design our treatment strategy based upon the kinds of the biomolecules in our body. So if you see in that context, then we can understand what is the meaning or any importance of this particular molecular diagnostic approaches. So it helps in analyzing biomolecules for disease detections. Then second one is your technological advancement. That means we know that there are certain conventional approaches in this picture. You can see liquid biopsy is a quite old form of disease diagnosis or cancer diagnosis. But now the things are changing day by day. Just precision medicine is another field in which actually you can see or you can analyze the say health status of a particular individuals by looking the presence or absence of a particular biomarkers in our body. Next, let's see what are the next generation sequencing and CRISPR-based diagnostics are all about in the next few slides. Now, all these things, they will lead to the third domain, that is your applications in the healthcare sectors. So let's see what are the molecular methods which are mostly used for pathogen detections as a whole. So you know that pathogens, they are also responsible for causing diseases which are not tense, which will not tense or which will not take you towards cancer, but simply they cause the disease and that disease will regress day by day. But some pathogens which are mostly they form cancer in the form of molecular approaches, like they used to integrate their genome, they used to integrate their own DNA and RNA materials with the host DNA or the RNA, and then finally they will be mutated, and those mutations may be very much vulnerable for these kind of health hazards. So if you see the molecular methods of pathogen detections, then there are few of the techniques that I have shown in this picture also. The first one is your polymerase chain based detection techniques. So all of you might have seen this machine that is known as thermocycler or polymerase chain machines. So what it does is that it usually amplifies a specific DNA sequences which enables for rapid detection and identification of the pathogens. That means if the pathogens are present in our body, so they will be having their DNA or the RNA materials inside the body. So the PCR or the primers that we have designed, that primers will be annealed appropriately with the target pathogens, pathogenic DNA or the RNA samples, and they will be amplified gradually. And after 35 cycles or say 40 cycles, what we can see in conventional PCR or the semi-quantitative PCR is that we can visualize the presence of the band in the gel. So looking upon the band, you can say the presence or the absence of a particular DNA of a particular pathogens in the suspected host. Now, second important category of PCR is that. So that's why I have written that polymerase chain reactions and its variants. There are so many variants of polymerase chain reactions. It will be like semi-quantitative PCR. It will be like a normal PCR. It will be just like a real-time PCR or nested PCR. So there are so many forms of PCRs. But the basic principles remains the same. Like you have to design the specific primers to identify or to amplify a particular DNA or RNA samples. So likewise, you can diagnose a particular. So these are the advanced form of the molecular diagnostic for any diseases. Now, second most important thing is that real-time PCR or qPCR. Mostly we know about the qPCR because that is nothing but the quantitative PCR. That means the quantity of a particular DNA or the amplified product can be measured. That's why they used to call it a quantitative PCR. It measures the amplification in real time. Real time means all of you know that from this diagram we can see actually what is happening with the DNA sample, our targeted DNA samples. This graph or the sequoia graphs exactly show that the DNA that we are targeting is getting amplified to a certain extent. And by looking the graph in whole, you can understand at which particular time or at particular cycle the DNA get amplified and what is the quantity of the applications or quantity of the amplification occurring in that particular time. So this graph plays a very, very important role in real-time PCR. That means in the current time or the process that is happening currently, we can know about the presence or the absence of the DNA samples. Then second one important thing in molecular diagnostic method is that CRISPR-Cas based detection systems. This is the picture which shows more or less we can understand about the CRISPR-Cas based detection system. These are nothing but the gene editing techniques which have been investigated, which have been utilized for diagnostic methods for cancer also. So in this particular diagram, you can see exactly a guide RNA is there, and this guide RNA is to take the other apparatus or the other enzymes or the other proteins near to the DNA, targeted DNA, and it is to cleave those particular DNA. Or in some cases, a particular probe will be attached with this particular DNA or the guide RNA. Now this RNA, when it get amplified or it get annealed with its target DNA, what it happens is that it will just produce some sort of fluorescence, and that fluorescence can be easily visualized under the microscope or under the, you can say, a particular apparatus. So that is the basic principle of CRISPR-Cas based detection system. Now, if it asks me exactly that what is why these kind of costly equipments are mostly used for, detecting a particular pathogens or present or absence of a particular pathogens, then I would say nowadays the things are changing very fast, very fast. You can say in the next, suppose you are doing a particular research on today, the next day you will be saving, you can see that your research is being directed towards something else. Okay, so time is running very fast. So in that context, actually we have to diagnose a particular disease up to 99% accuracy. So that is very, very important. Or else you can just take a small strip of paper and that paper, a small strip of paper also can diagnose the presence or absence of the pathogens on that. But then specificity and sensitivity will differ in that kind of a diagnostic methods, which are mostly conventional in nature. Now we'll go to the third part, that is biosensor. So what are biosensors? These are the devices that incorporates biological materials, just like your biomolecules, maybe proteins, antibodies, or any enzymes, to detect the presence or absence of the pathogens. Why I am telling presence or absence of the pathogen means sometimes we used to carry out so tedious process, but finally we will not get single presence of particular pathogens in our suspected samples. That's why I am telling that presence or absence of some pathogens in that. So this picture actually depicts the form of electrochemical biosensors. So these are the components in this electrochemical biosensors. One is your bioreceptor interface, then transducer, and after that it used to send these signals to the amplifier, and amplifier it sends signal to the signal processor, and finally on the screen you can clearly visualize the interactions between the biomolecules happening over here, but you can see whether or not the interaction is happening the way you want it to happen. So this particular device is an example of electrochemical biosensors that detects the pathogens based on changes in the electrical signals. That means when a particular pathogens, specific pathogens, they find their counterpart, just like in this picture you can say maybe these are the antigen or antibody, and they are having these conjugate molecules or its receptor molecules on that. So some molecules will directly interact with that, some molecules will not interact. They will just sit on their surface. When you wash them out, then they will simply clear out. So in that process, you cannot say that they are reacting or they are having any kind of relation between their ligands. So in that case, actually, you can directly say that whether or not a particular reaction is happening on the surface or on the chips, you can say. So based upon the change in the electrical signal, that means what kind of signal change is happening, volt in the sense of ampere or in point of voltage, difference will be there, and that will be detected on the your display systems. Now, second most important category is your optical biosensors. In electrochemical biosensor also, you will be getting 10 or 15 different kinds of biosensor devices. Likewise, in optical biosensors also, you will be able to get say 10 or 15 kinds of different optical biosensors. But try to understand the meaning of these optical biosensors. It, it is having the optics principle of optics. This picture shows about the biosensors device based upon the optics or physics, you can say. So that helps in detecting pathogen based on the changes in the light signal. In this picture, let me try to explain the phenomenon actually, what is happening. So there will be one incident line, and that incident line falls upon a particular surface, and that surface will be inert, or you can say that is made mostly that is made up of gold film on the glass cover sheet. That is inert. So that's why this kind of materials they are used for sphere based or optical based biosensor techniques. Now let me explain it in the little bit of detail about the optical biosensors technique. Now when a particular wavelength falls on this particular surface, what happens? Either it gets transmitted to the other side, or it gets reflected back in the same directions. But this angle, this angle plays a very, very important role. Now, this depends on this angle. Sometimes it becomes bigger, sometimes it becomes very small, sometimes the light also can reflect in the direct way to the 90 degree to the base. So in that case, what you can say that there are certain changes happening on this particular surfaces. So in this picture, what is happening actually? Antibody, green molecules are antibody, and this pink or the violet molecules, they are antigens. So if the antigens are specific to the specific antibody, then there will be a strong binding between the antigen and antibody. And if the molecules are not specific, then simply what will happen when you made to flow some sort of buffers on this channel. So what will happen? They will simply go out, and all these things will be visualized like this screen will be there, and you can visualize in the form of graphs in that. So the more molecules will interact on the surface of this biosensor or chip molecule, there will be changes in the incident light and reflected light, and this theta angle will be changed. So based upon that, all the data they will be gathered on the monitor, and they will just try to show you that okay, this is the kind of reaction happening over there. So based upon that, you can interpret your results, and you can interpret the interaction between different biomolecules that is happening on the channels, just like this. So this is a kind of, say, you can biosensor, and nowadays mostly this is used because within a second or within a minute you can say whether or not two molecules are interacting efficiently. Then third category, your mass-based biosensors. So there are also different types of mass-based biosensors. It detects pathogen based on the changes in the mass. I have not shown the picture, but thing is that, so when they interact, actually there will be difference in the mass, or there will be degradation of the mass after interaction, or the mass will sometimes increase. So based upon that, the biosensor says that okay, there is something happening under that, and we'll analyze taking some sort of algorithms or some sort of calculations, and then you can say that, okay, this is what is happening. Now other techniques, like there will be lateral flow assay, and all of you know that more or less we have suffered from diseases like malaria in our lifespan, sometimes in past also. But from that we can, so, or if you might have seen a small device, that is pregnancy diagnostic kit, that is a strip based kit. So what is that? This is a kind of very simple device to see, but thing is that when you work on these kind of devices, it has to be 99% specific, or else when we work on a particular device like diagnostics based upon a disease, there is no estimate. If your diagnosis goes wrong somehow, then suppose in place of malaria, you can you detect wrongly this presence of the disease, and you say something else. In that case, so many things will happen, and then you may drag to the court also. So for that, actually, this thing seems to be very, very easy. But when you practice these things, or you try to develop some kind of techniques or some kind of assays, it becomes very tedious and it becomes very difficult. But anyhow, as a scientist, we have to practice those things, and it is our duty. As a biotechnologist, we have to do anyhow. So this one is your lateral flow assay. That means the something that is flowing from one side to the another side. So that remaining says like that. So meanwhile, what happens actually in this particular strip or in this particular paper, you can have some molecules so that they will interact and they will produce the results in forms of some bands or some colorful bands, or sometimes if they do not react, then they will not produce any kind of bands on the membranes. So it is a rapid point of care test that uses antibodies to detect pathogens. From this, actually, you see, this is this you're putting a drop of blood on that, and that drop will travel towards this direction because of the capillary movement of the paper. Now the conjugate pairs are there, and this is a see. Actually, this is a very interesting device nowadays. Almost everywhere you go, say when you talk about development of a particular test based upon lateral flow test, they will say whether or not there is a feasibility for developing a diagnostic based upon lateral flow immunoassays, antigen antibody interactions, or complement fixation interactions, whatever. But the thing is that it is very cheap and easy to use also. Suppose you just train your local technicians about this, they will go to the village and they can detect the presence or absence of a particular pathogens. Now, it mostly these are nitrocellulose membrane papers. All of you have practiced some sort of experiments with using nitrocellulose membranes because they are more porous, not you can say they are not so reactive, but they are inert in compositions. Okay, based upon that, whatever the reactions you do, you can visualize and you can also store. You can also store them for at least for 2 years. It's such. That's why Western blot and other things they are practiced or they are done on nitrocellular membrane papers. So these are very important things that nowadays to develop some kind of diagnostic based upon the lateral flow assays. Now, one thing I just forgot to explain, I was just forgotten to write over here is that it is based upon the interactions between the antibody and antigens which are on this particular test line, right? So in this case, particularly, we are going to detect the presence of a pathogens over here. So we have to put a line or immobilize the certain antibodies which are present, which are just specifically designed against these pathogens on this particular line, so that when they will travel, if they are specific, they will bind. If they are not specific, they will not bind. And after their tight bonding occurs, they are again like labeled with some fluorophore compounds, so we can produce certain light. Just like you can see in LSA, what happens? The primary antibody and secondary antibody play a very important role. Likewise, you can have one line on that strip paper. You can say, okay, this particular presence of this antigen is there in a suspect pathogens. The next is your enzyme-linked immune solvent assay. This is a quite conventional and old techniques, but this is very important. Nowadays, or you can say in college level also, to make them understand what is happening between the antigen and antibody. If they are specific to each other, they will react and they will produce some sort of colors. If they are not specific, they will not produce any colors. Simply, you will get the color of the dye, and that based upon the readings or based of the optical density, you can say, okay, some sort of reactions has happened over there. So in this picture, I have depicted both forms of the ELISA, their direct ELISA, indirect sandwich ELISA, and competitive ELISA. In picture, you can understand, you can see actually there will be antigen and antibody, and these antigens, if they react with these are tightly immobilized on the strongly immobilized on the surface of the, in this particular, you can say 96 well plate. Now, if the antibody, they are specifically designed, or sorry, they are specific to this particular antigens, they will bind on this, and it will again bind with the secondary antibody, which are commercially available, and the secondary antibody has been labeled, or it has been, there is the association between the secondary antibody with uh, enzymes. So this is the enzyme. So when this enzyme reacts to a particular substrate, it is used to produce the color substance or color. Now, based upon the color, you can say that that enzyme is reacting or the antigen is reacting with this antibody. So these are some of the conventional techniques used for the pathogen detections. Now, let me remind something, like when we talk about pathogens, then we are talking also about the antigens which are present on the surface of the pathogens. Now, pathogens in the first slide, as I said, that the pathogens are so many different pathogens like bacteria, virus, prions, proteins, or simply proteins, or you can say algae or fungus. So they are having different morphological structures. So their structures are quite different, and then the antigens they are having or they are interacting in the body are also quite different. So based upon the antigens, we never know that our body's immune systems is reacting in a different perspective, what way that is going to react, and what antigens they are having on the surface also we do not know. Okay, so when practicing this kind of diagnostic tools development, we'll have to understand the nature of the antigen first. That is quite important to understand the nature of the antigens, and then the development of the antibody against that is a secondary issue. But understanding the structures of the molecular structures in a fully you have to understand the structures of the antigens. Bioinformatics also you have to apply it in some context, and sometimes you have to do wet lab experiments also. Okay. Now, okay, there are a few advanced form of diagnostics which are based upon. The first one is your immunomagnetic separations, or short form is IMS. In this particular picture, you can understand what is happening actually. No? So it uses magnetic beads coated with antibodies to isolate pathogens from a sample. This particular well, or this is a container in which actually there are some bacteria or the pathogens, and those pathogens have been, they have been embedded, or some sort of magnets have been attached with this pathogen, and that those magnets, those particular beads will be with the help of a particular magnet, they will be attracted, or they will get attracted towards the particular magnetic beads, and will be able to separate this affinity, the molecules which are having the affinity towards the magnets. So this is a very beautiful form of, but to some extent it works. Okay, we cannot rely upon a particular techniques for all the diseases. There are different techniques, there are different assays, there are different tools for diagnosis of various forms of diseases or pathogens, we can say. Phage-based methods. All of you know that what are the bacteriophages. This picture is for this particular line. Phage-based methods, that is also, you can say, that is also used for some specific purposes, like viruses that infect bacteria. They are called as phage, that help in detecting diseases. How they help in detecting diseases? From this picture, probably I will be able to make you understand. See, in this particular picture, actually, the phages, they are trying to attack a particular group of virus, or say E. coli. All phages that do not infect all bacteria, phages they are having specific for specific bacteria. Just like they are having specific receptor based upon the presence or absence of receptors. The phages they used to come and sit on the bacteria. Now, when this particular phage they enter into the bacteria, they will be having, they will try to replicate themselves, and when they replicate, and they finally what will happen, the bacteria they will get lysed. Now, based upon the DNA sequences, or DNA sequence, or the RNA sequence of this particular phage, we have designed our real-time PCR. we have designed our primers, so when it gets lysed, so easily we can say that this particular say phage has been this particular bacteria has been infected with this particular phage. That's why there is an amplification, and we can say, okay, this is the case, or there is a presence of this particular bacteria in the infected individual. And based upon that also we can design some immunoassays or lateral flow immunoassays also. Now this particular picture depicts actually when a particular bacteriophage infects a bacteria, and this particular bacteriophage is engineered in such a way that when it infects, it try to incorporate its own DNA or the RNA material with that of the with the infecting bacteria, and finally they will produce some sort of luminescence or the fluorophore that can be detected using a particular say this one monitor systems. And the third one, you can say that they will be immobilized on the particular surface, and they will be when there will be interaction will be there, and you can visualize their interactions using a particular microscope, a particular monitor. Then so, these are the techniques that we have to utilize. This is the basic principles involved in that. So you can use your own concerns, you can use your own field, and you can use the techniques that just now we have discussed, all starting from the say lateral flow based immunoassays, immunomagnetic separation techniques, phage, and AI-based diagnostics. Now you can develop our own diagnostic tools. And then next one is your most advanced form of the diagnostic, that is AI or artificial intelligence powered diagnostics. So this AI can be used to analyze the complex data and identify pathogens more quickly and accurately. This is a picture just to make you understand what is AI, and nowadays AI has very important role. So mostly what this AI does is that it used to analyze the data using all forms of algorithms that it has, and then finally the all data will be concluded, all data will be interpreted in such a way that it can say the presence of a particular pathogen in the body. Now, I just now discussed about some of the important forms of the diagnostic or the tools of detecting pathogens using certain techniques. There are so many other techniques also. Probably the next speakers, those who will be giving speech, they will be discussing in detail about different techniques in detail. So I just thought that let me just make the introductions very simple so that in the next few slides other speakers can speak in a much more detailed way about the content. Now, cancer detection innovations. So what new things, our techniques or our FDP says that next generation molecular diagnostics. That means whatever the conventional techniques we are having, starting from the biopsy material detections or say PCR bed detection methods, they are getting older nowadays. Day by day, they are getting older. So what next can be done regarding the diagnostic methods for the pathogen and cancer detections? So that's why it says that cancer detection innovations, starting from the liquid biopsies. Like what is a liquid biopsy? You see in the particular picture, you can say it is a method for detecting a circulating tumor DNA in the blood. Most conventional method of detecting pathogens or presence of cancer-causing pathogens. So simply you can collect blood from the human beings or suspected pathogens, suspected individuals, and they will be analyzed for the presence or the absence of a particular tumor DNA. That means, as I said somewhere in the primary slides, that we do not know what is the cause of the pathogen, cancer, what is the cause of the cancer, or what is the primary source of the cancer or origin of the cancer. Now, when those cancer cells they metastatize from one place to another places, we do not know that what, in which place that particular cancer cells is going to reside or going to re-establish itself. It may be lungs, it may be heart, it may be brain, or it may be some peripheral tissue like fingertips. We never know exactly, right? So that's why liquid biopsy means the blood will be collected and the presence or absence of a particular pathogens or that particular DNA materials out which has been released from the cancerous cells will be detected. So it detects cancer and determines the treatment options. See, what is said determine the treatment option means if the number of the say circulating tumor DNA or the tDNA, the number of the tDNA is more in the circulating blood, then we can say that the process of metastasis is happening very fast and it may go to any places in the body. That means based upon that, the doctor can determine the treatment options. And then it monitor the responses for the treatment also. So that is what the liquid biopsy is all about. So though this is a old technique, or you can say conventional techniques, but then we cannot just ignore these techniques because that is very, very important to know what is happening in our body. So unless we collect blood, unless we collect the biopsy materials, everything cannot be told by AI or the artificial intelligence. So, cancer is a very, very dangerous disease. All of you know that what is happening. Although we are taking all sort of preventive measures to not to happen cancer in our lifespan. But see, as a life scientist or a biotechnologist, we know that if we are playing some sort of mutations or there is a mutation happening in our DNA or the RNA or the genome, anyway, we never know what kind of mutation is that. So based upon the nature of the mutations, the cancer or the say development of the cancer depends mostly. Okay, now the second one is your molecular profiling. It helps in profiling the say you can say parameters also. So it helps in advancement of the sequencing, which tailors the therapies to individual cancer types. That means when we are coming across diagnosing a particular pathogens or a cancer cells which are correlated with pathogens or the primitive cause, the primary cause of the cancers may be pathogens also. In that case, it is very, very essential for molecular profiling of the cancer types or that particular, say, host or infected host. Based upon that only the treatment can be designed. So the third important things is that biomarker discovery. All of you know that what are biomarkers? Biomarkers are nothing but the biomolecules, just like your DNA or RNA or proteins. Any molecules in our body, you can say they are the biomarkers. That means their presence tells about the presence of the cancer in our body. Just like these are some of the biomarkers, just like miRNA or some sequence of the DNA, proteins or cellular metabolites, exosomes, organic materials, anything can be biomarkers. Not that DNA or RNA sequence, anything can be biomarkers for that. Now, the cancer study fields, it determines that new biomarkers also day by day they are doing very advanced form of research which tells or which finds out different biomarkers we can tell about the targeted therapy and for the early detection of the cancers also. Now, I have not kept the PowerPoint presentations in detail or in detailed form because there are so many different forms of diagnostics available nowadays. So their working principles are more or less similar. Their applications are quite different, and then the cost also quite different. So there are so many experts in this field, so they will be talking on that in detail. So just my duty was that just to to make you aware about the vulnerability of the cancer and then the pathogens which are associated with cancer are also very, very important, because by knowingly or unknowingly we do so much of things which leads us towards the formation of cancerous cells in our body or the modification of the cells, and that they will get metastasized, as I said earlier also, that once that process goes and that we never know that what is happening in our body, and then also finally it becomes too late for us to. So just to understand that, you have to go through different materials and make the students aware about, the different forms of the diagnostics. Now, if you look at the limitations and challenges of these particular techniques, there will be challenges like sensitivity and specificity. As I said, that there are so many different diagnostics based upon the optical methods, based upon the electronic methods, based upon the just conventional method like physical or chemical methods. But once the sensitivity they are not defined and the specificity they are not defined, that that product cannot come to the market. And we are not talking about the product, so we're just trying to understand what is happening, right? So for that, actually, sensitivity and specificity of a particular tool has to be, and for me, that has to be just more than 95%, so that you can say, yes, this is malaria, yes, this is brucellosis, or any disease you can tell in 100% or 95%. It's quite good sensitivity and specificity. So next one important thing is that cost and resources availability. So high cost of the advanced technology limits accessibility and lower resource setting. That means we have to design our tools or the diagnostic tool in such a way that the cost should be quite less, so that the general people or the say village people also can use the techniques in much more easier ways, and that also depends again on the sensitivity and the specificity. Okay, so these are quite very important, three important fields on which actually the diagnostic or the fate of the diagnostic tools or the technique depends on. Now third one is your data interpretation and integrations. Now and the complexity of the data from molecular diagnostic requires a skill. Of course, that requires a very skilled personnel for data interpretation, because there are so many algorithms nowadays, mostly to study the structure of the antigens or the study the structure of the proteins. We have to rely upon bioinformatics tools. All of you know that bioinformatics depends on bioinformatics rely upon algorithms, so many algorithms to predict the structure also. So for that, actually, one skilled personnel is very essential for interpretation of the data. Now, I'm just coming to the end of our today's topic. That is, what will be the future directions of our pathogen or the diagnostic tools? It will directly say it helps in nowadays we have to rely upon artificial intelligence. Actually, the more we will be engaged in techniques, the new techniques are coming in the market day by day, and those techniques will be having their own importance. So that way, actually, AI has to be integrated in our diagnostic techniques or diagnostic development. So utilizing AI can enhance data analysis for diagnosis. Now, these things will be directly applied for field like point of care testing. So point of care testing is nothing but developing a particular diagnostic, which will be very simple in the sense that just imagine you can say you can have a small diagnostic very sensitive, and that is also very specific in their form. So that has to be development of portable devices for rapid results and accessibility. That is a quite important thing for diagnostic assay. If you are diagnosing a particular disease with 1,000 rupees, and if you are diagnosing the same disease with 100 rupees, definitely it differs. And there are some, you can say, anyway, people they will try to prefer the 100 rupees diagnostics. But then what about this sensitivity and specificity? So we'll have to focus mostly on this kind of point of care testing methods or point of care testing tools for diagnostic development. And then we will have to also to do some collaborative research initiative, which is essential for advancing diagnostic innovations, because you know that there are microbiologists, there are surgery specialists, there are doctors, there are botanists, and so many things are. I mean, so many professionals are there. So once you'll be having collaborations with different fields, chemistry, physics, and all those things in the university level. I'm in the university, so I'm talking about university level. So it is quite essential to have a collaborative kind of things, collaborative research among us. So that will help, diagnose, developing a new kind of good diagnostics, a new kind of good assays, which may in future will be helpful for designing a good set of things for eradication of a particular disease. So to conclude our talk, I will just give you 3 points. One is this next generation diagnostics, improved pathogen detections. So it helps me enhancing the diagnostic development or enhancing the detection methods. A better point of care will be there, and future considerations like there will be ethical considerations which will shape the healthcare industry in our country. So I think with this, I would like to thank all the participants once again. Actually, I'm not feeling a little bit good because of my health is not good right now. So for that, you can just tell, okay, anyhow, we have to understand, we have to have the concept at least in our mind so that we can translate it to our students, or we can translate it to our society day by day. So if there are some sort of questions should be there, then I'll be quite happy to address those questions also. Thank you once again.
Thank you, sir. Thank you for such an excellent presentation and cover the topic with overall idea how to go from conventional diagnostic to advanced method. It was very insightful talk, sir. I am sure from today's session, our participants must get overall knowledge on molecular technique and its applications in current health science. So now I am requesting our participants to ask some questions. You you can ask your questions by raising your hands or by using chat box. Okay. Anybody? Yes. Dr. Sir, can you take him on mute? Dr. Khora, please. Khora sir has raising. Both can unmute your mic. Khora sir. Dr. S.H. Khora. Hello.
Yes, sir, please.
Dr. Khora and Dr. Mousim has raised their hands.
Dr. Khora, can you just unmute yourself? Actually, we are unable to
Listen.
listen to you.
Look, there was a question from Dr. Mosim Misro. That is, can NGS help in real-time outbreak surveillance and antimicrobial resistance tracking?
Okay. Okay, right. Definitely, sir. Let me take back this one. Am I audible?
Yes, sir.
Yes.
Yes. Right, good. So actually there are two things. One is your next generation sequencing and then antimicrobial resistance, right? So next generation sequencing, it's a kind of sequencing in the sense you can sequence a little bit small fragment of a DNA materials or a small fragment of a gene, or you can secure the whole genome also. Okay. So by doing that particular process, but that is quite tedious process. You know, as I said, that one needs a very technical or expert technical person to analyze this sequence. So it will be a little bit of tedious. But then what happens? You know, those are quite accurate process. You know, molecular diagnostics or next generation sequencing is a quite accurate process for diagnostic almost all forms of pathogens in the body. So, but thing is that you have to design a particular primer for that, and then when you are going to analyze those sequences, you have to compare your sequence with some other similar sequences, and then finally you have to come up with your results. So definitely next generation sequence plays a very, very vital role in this particular diagnostic era. And regarding the second question, that is diagnosis of antimicrobial resistance group. So we have to understand what this antimicrobial resistance are all about first. That is the most important questions that we have to address. And once we understand what is it, that means which group of microbes they are getting resistance in the body. Which groups of microbes are getting resistance in the body, and what is the basic physiological mechanism happening in the body, that we have to understand. Then by the techniques like just try to find out the conserved sequence of those things. Probably sometimes we will go to literature review also. You have to go back to literature review sometimes, and then from those literatures probably will be able to get the sequence or the genomes or the genes which are conserved in nature, and those genes need to be PCR amplified, sequenced, and then finally you can come to the conclusion of differentiating between the this antimicrobial resistance group of bacteria. Definitely, there are two important things to keep in mind. Once we work on microbiology or the genomics or the proteomics, definitely there are so many things, because see, it looks so similar. I mean, easy for sometimes to develop a diagnostic tools like lateral, because I was just working on lateral flow immunoassays assays. But when the sensitivity and the specificity comes, then definitely we just put our hands over just we cannot help them. So that is what I can give you the assurance that we can do. But once the instruments are there with us and the technicals or the expertise are there with us, we can do a better development also.
Thank you, sir.
Thank you very much. Thank you very much.
Thank you, sir.
One more question was raised by Dr. Sobhagini.
Yes.
He just wanted to know whether this healthcare cost will really reduce in long term.
Actually, sir, this is a quite big and very important questions. But who will be responsible for this? See, the company, they are taking the techniques, and I mean, they are just putting their intellectual property, and they are just investigating those things in different forms. So, but as I was working on 2 or 3 important diseases, just like brucellosis, anthrax, and one form of malaria, I know that. Yes, so I know that what are the importance of lateral flow based diagnostic in the context of all the states of India. So all the medical personnel, they to the village level. Once you go to the village level or the panchayat level, or if you go to tribal places also, so the people those who are they are quite ignorant about the disease and all. But only the thing is that once you develop a very small diagnostic kit based upon just like a small point of care test, or you can say a little prognosis, they are quite ready to give their samples also for diagnostic assays. I mean, you can diagnose those things in a very simple manner also. So in near future, we cannot say that health sector, you can just reduce the cost of the drugs or reduce the cost of the diagnostics. That depends on the form of the disease in particular. So I can say that only.
Yes,
Thank
that's
you,
right,
sir.
sir. Sir, can you take up 1-2 more questions?
No problem, sir. No problem.
This question was from Professor S.S. Khora. He
Yes.
just wanted to
Yes.
he just wanted to know whether is it possible to diagnose the cancer stem cell by existing methods.
Of course. Of course. The answer is really there, sir. Actually, once we find out the biomarkers, specific biomarkers on the cancer cells or specific biomarkers present on the cancer stem cells, then definitely you can find out or you can differentiate between the stem cells for the cancer and the cancer or the cells which are circulating in our body in presence of the cancer. Okay, that we can do. But then I do not know what sort of ethical issues will be involved in that. So that is sometimes we create problems for that. But anyhow, we can differentiate between the stem cells and also differentiate between the conventional cancer cells. It is possible, sir, quite possible.
Yeah. Thank you. One more interesting question is from Dr. Rudraksha Panda. He just wanted to
Yes.
know which molecular diagnostic techniques are to be known the best methods for cancer detection according to you. I think it
According
will
to
be
me,
the,
yes,
yeah.
definitely, because we are not doctors. So anyhow, we can assume and we can just tell few things like whatever we have understood about the cancer. When you go to the hospitals, what they used to say, they used to physically diagnose a particular cancer tissue. They used to try to know that what kind of cancer is there in our body. Physically, they used to try. Then next one is that they used to isolate the biopsy material, and then in those biopsy materials, that they used to find out, they used to find out the presence or absence of the biomolecules or say biomarkers in that. So real-time PCR will be quite sensible and quite important, methods nowadays to diagnose or presence or absence of a particular cancer. I think, according to me, that will be the quite appropriate techniques.
Thank you, sir. One more.
Great. It's great. It's great. One more question was there from Dr.
Yes.
Diptya Sethi. He just, I mean, she wants to know whether uh, diagnostics used in infectious diseases outbreaks early cancer detection. In the sense, can a common man know whether he or she is going to be infected with cancer? Are there any symptoms or maybe early what is called human detection methodologies which are available so that this thing can go to the society, that these are the provocations or methods will be used so that can go early and detect the things.
Yes. That's a very important question actually. When you involve society as a whole, no, that plays a very, very important role. Whatever you are doing, either you are no, developing a diagnostics or you are developing a vaccines or anything that finally going to society only. So for that, actually, literacy plays a very important. Once we go into the society, you have to make that aware about what is the fate of the cancer, what is the fate of the cancer as a whole. So and not just by saying that okay, if you consume tobacco, then you are going to suffer from cancer also. You are going to, if you are consuming alcohol, you are going to suffer from cancer. It's not that there. Sometimes, if time permits, I mean that is what the practice that I used to follow most of the times. I used to have some sort of pictures on my mobiles and sort of pictures in a bag also sometimes. So while going to society and people, those who are quite ignorant, and you observe that they are involved in such kind of activity that may tend very quickly to take them towards cancer also. We have to make them aware. First, this is the primary stage. I mean primary stage for that purpose. Then second one is that once we develop any kind of diagnostics, then that has to go through a particular channelized, that have to be channelized properly to develop, to have it certified from the governing body or the say government. So you have to take many things into account when doing working on cancer or developing a diagnostics for diseases. Did I answer your question, sir, or something else was there?
Yeah. But it was fair enough. Fair enough. I think a lot of things have come up.
So when you are talking about society, then definitely, sir, we have to involve directly or indirectly with the society. We have to go to the society people. Sometimes we have to take the sarpanch also, the head of the village also, to aware them about these problems that we are facing. Or else what is happening, sir, whatever the things will happen, that will simply go into the waste. Only that, what I can say about it.
Yeah, that's great. In fact, almost all the institutions involve their students for various extension activities, and the
Yes, yes.
student goes to nearby villages, nearby societies. Even they are being connected with the family. If they know these sort of things from the experts or the researchers who have great proficiency in this domain, it will really solve some of the content that will help that to the society.
Definitely, sir. We never know actually. It is going to solve some of the problems or most of the problems we never know. That depends on the attitude of the students actually. So the way we take them to a extensive activity or extension activity, or sometimes we have to take them to a just like say Bakti Cancer Research Institute or some research where actually these kind of things are going on. So out of 100 students, probably 5 will be take their keen interest to understand the concept, but to understand what is happening actually and what is their societal role and all these things. So as a faculty, I think this is our primary goal or intention to to do this kind of activity in our institutions.
Fine, sir. I think we do not have more questions in Q&A platform.
Sir, actually,
Yes.
one or more two faculties raised their hand. I think Dr. Rajesh Sahu. Rajesh Sahu, have you any question? Dr. Rajesh Sahu, you can ask by unmute.
They also can put their questions in that message box, chat
Yes,
box, so
yes,
that
sir.
we can escalate the questions to Dr. Maji.
Okay. So I think most of the questions I cleared from my side actually.
Yes,
Maybe I
yes,
can do it.
Yes, sir.
yes, sir.
So maybe answers will be different for different research person. But for me, we can do many things. But thing is that we have to be involved in that activity to the maximum extent we can be involved. That is what we can do.
Fine, sir.
But I'm
Over
quite
to-
happy. I'm quite thankful to you at least that you have started the faculty development programs, probably other programs also. We know that meets many of the times. There are different from the different forum we can discuss those things later on also. But then at least these kind of programs helps in energizing the faculty and the participants in many ways actually, sir. These topics nowadays, we know that cancer is a problematic issue, pathogens is a problematic issue. But then time to time, we are just not getting involved 100% into those things, and just forgetting every day. We are just doing same kind of mistakes in my home also. I observe that, okay, doing the same things, and probably who knows that what is going to happen with the health of body. So we have to be little bit conscious why to go for cancer actually. So for that you have to adopt some of the healthy procedures, not to happen cancer, not to infect with this kind of pathogens in future. That is what I can advise to all the youngsters.
Thank you, sir. Thank you so much. Actually, this is a very emerging topic, what I want to add. And two things, I think, what you said that, sir. One is to early detect the pathogens for the cure of the cancer, and another thing, the antimicrobial resistance, to check the antimicrobial resistance. For these both of these emerging subjects, we can rely on that molecular diagnostic. That molecular diagnostic is, I think, and molecular diagnostic will be the one of the pioneer diagnostic method for doing this type of things, and we can get the early result. And and also you have also mentioned that the specificity and sensitivity is high in that molecular diagnostic method. So it will be better if you go. And one thing, sir, we have also added, and one of the our participant has asked that question, that the cost of the molecular diagnosis. If the government will go and do something for that to reduce some the cost of the molecular diagnostic kit, then that will be implemented and in the health science for a regular basic test. And with that, we can get early detection and early diagnosis. That but we know the proverb, prevention is better than cure. Like that, early detections will be better than go for the ICU. Like that. Um, these things. Thank you so much, sir, for your valuable times, and you have given your response within few short period of times. And
I am actually ready, 100% ready in all whatever the times you can call me, no problem. But
thank you
that
so much,
depends
sir.
on the availability of the, you can say,
Yes,
other faculty members in our department also. But anyway,
And sir,
I'm completely,
we are waiting on
yes.
Saturday to you.
yes, definitely I'll be there, sir, no problem, sir.
Thank you so
Thank,
much, sir. Okay,
thank
thank
you very
you
much.
all for your active participation, all the faculties for the active participation. We are now going to close the today's sessions. We'll inform you about the next day program in the WhatsApp group soon. Sir, right, sir? Can you close
Yes.
this session?
Yes,
Yeah,
please.
of
Yes,
course.
please. With
Ah,
permission from the
okay,
principal.
fine.
Okay, sir. Thank you, sir. Thank you. Now I am closing this session, and we'll meet tomorrow. Thank you.
Thank you.